So this study, we focused on patients with transformed lymphomas, so transformed follicular and transformed CLL into Richters. We felt like in these two diseases, patient outcomes were still poor, especially Richters. And so we were focused on trying to modulate the immune microenvironment with a PI3 kinase inhibitor, Copanlisib, and then combining that with Nivolumab in the hopes that the two would synergize together and get better upfront response and then better prolonged response. What we found was that in Richters, there was a small subset of patients that responded, and those patients that did respond tend to have prolonged response. So it mirrored kind of similar to what we saw with single-agent Nivolumab, maybe a little bit higher response rate. And then on the transformed follicular side, the response rate was much more promising, above 60%, but the duration of response was shorter. So it seemed more like the Copanlisib was more effective in that setting. We were able to do some correlative work looking at patients who responded and patients who didn't, and we did notice some significant differences in their genetic signatures, especially for T regulatory cells. So it seemed like in non-responders, the inhibitory T cells were a lot more abundant and more active.